Hi, everybody. My name is Josephine, and I'm very excited to be here with you today to celebrate the International Day of Women and Girls in Science. This is session number 2 of our event that will be followed by session 3 at between 7 and 9 a.m. tomorrow on the 13th. The time is in GMT. So if you have not heard about the International Union of Forest Research Organizations before this event, let me tell you a little bit about it. It is a non-profit, non-governmental global network for science cooperation. And it allows the collaboration between around 15,000 scientists from about 630 member organizations from 115 different countries. And within IUFRO, science is divided between different divisions, and that include Division 7, which is organizing the event today. Yeah, and that focuses on forest health. Division 7 is then divided into two research groups, forest pathology and forest entomology, which are then divided into working parties that tackle many different topics related to forest health. So in the future, if you would like to get involved in IUFRO, you can directly contact our office holders that are studying topics that relate to your personal science. You can participate in IUFRO co-sponsored events that include conferences, meetings, and webinars such as the one that is organized today. You can read and contribute to IUFRO publications, communicate and publish news in IUFRO media, and even become a member and become an office holder. So the big event for us this year is the IUFRO World Congress that will take place in Stockholm in Sweden in June of this year. And for this meeting, Division 7 office holders are actually organizing as many as 15 different sessions. So it will be a very busy and interesting schedule. So I would like to take a moment to thank the members of the organization committee. They are Irene Barnes, Quentin Guigna, who will be our moderator for today, and Jeremy Allison. And I would like to give a special thank you to Prabina. She is a GIS technician at Clemson University in the USA. Her skills, time, ideas, motivation, and enthusiasm have been sincerely appreciated when organizing this event. She was moderator for our first session, and hopefully, if she can join us, she will be presenting her work today during session 2. So let's go through a few housekeeping items before we start. Quentin will leave his camera on, so it is a bit more lively for our speakers, but please leave your cameras off. This is to preserve bandwidth. You are welcome to use the Q&A feature to ask questions to our speakers. Quentin will then ask them on your behalf. You're also welcome to use the chat at any time for any other forms of communication. This includes alerting us of some connectivity, sound issues, or to say hi to your colleagues, announce future events, etc. And you might have noticed that we have a break room during this event. So the point of the break room is that the speakers, after giving their own presentation and answering a few questions from the audience, will then move on to the break room, where you can have small group conversations with them, ask further questions, and introduce yourself. And there is no rule about how long you can stay in the break room. You can stay there for the whole of the event if you want. If you are worried about missing some of the presentations we have for you today, the meeting is being recorded, and it will be posted on YouTube after the event, and we will send you the links. So before we move on to our first speaker, I would like to wish you a happy International Day of Women and Girls in Science. And I would also like to say that we are here to celebrate the women that have come before us and empower the women that will come after us. Before we hear from our first speaker, I would like to take a moment to reflect on and acknowledge why we're here today and why today is important. So first of all, I would like us to remember that we cannot fight for women's rights without fighting for the rights of all women. I mean that while we fight against sexism, we must also fight against other systems of oppression that oppress women of color, indigenous women, disabled women, and members of the LGBTQ+ community. Second, our community, which is the environmental sciences community, is approaching parity, and in many instances, women even outnumber men. But it is important to remember that representation is not the end game. In fact, it is only a stepping stone towards a community that is shaped by a diverse community of people and that serves a diverse community of people. It is a community in which every voice is equally respected and valued. Finally, I would like to acknowledge the women that are not in the room. I am talking about the millions of women and girls across the globe that are being robbed of their basic human rights, including education, because of the occupation of their homeland, human trafficking, child labor, child marriage, and systemic racism. So let me end this introduction with a quote from the Black lesbian activist and poet Audre Lorde when she said, "I am not free while any woman is unfree, even when her shackles are very different from mine." And with that, I would like to wish you a happy International Day of Women and Girls in Science, and I will let Quentin take over and introduce our first speaker. Thank you.
Hi, everyone. Josephine, thank you very much for this very inspiring introduction. Let me echo you and say a happy International Day of Women and Girls in Science to you all. I saw Prabina just arrived while you are giving your talk. So, Prabina, are you with us? If you can switch on your camera and your mic.
Yes.
Yes. All right. So our first speaker, Kimberly Wallin, Professor Kimberly Wallin is not with us. So we'll start with Miss Prabina Sharma. Miss Sharma just submitted her master degree at Clemson University last year in South Carolina. Her master was entitled Study of the Morphological and Physiological Properties and Pollinator of the Invasive Calorie Pair. And Prabina is now working as a GIS technician. So Prabina, if you are ready for it, the floor is all yours.
Okay, thank you. Okay, let me share my whole screen. Can you guys see it?
Yes, we can.
Yeah, we can see.
Okay.
Awesome.
Thank you, IUFRO, for this amazing opportunity to share some of my research finding and to learn about other people's research finding as well. So this is a part of my master's thesis where we studied about the pollinators of the invasive calorie pair in the Clemson University. So I would like to start my presentation from the introduction of what this tree actually is. So calorie pair is scientifically known as Pyrus caloriana and belongs to family Rosaceae. This tree is a non-native invasive tree in the USA. However, it is native to the East and Southeast Asia, and the amazing thing is that it is the endangered species in Japan. So this tree is medium-sized, fast-growing tree species that can attain the height up to 35 feet, and it has a beautiful pyramidal shape because of which it is considered as an ornamental tree here in USA. So this tree has a white flower in cluster of 5 to 20, and the flowers are hermaphrodite, protandrous, and diploid, meaning that they have different male and female reproductive organs, and they are easy to adapt to the new environment. And the seed bud dormancy of this tree can last up to 11 years, making its seed bank like highly across like a range of years. So we can, if the tree seed is present there, it can still be reproduced after 11 years, so which is a long period of time. So the distribution range of the tree is, as you can see in the picture here, like all the dots like is present all over the US. However, the dark color shades represent the state where it is present in higher number, So after being introduced to US in 19,000 by a scientist Frank Ian Mayer to fight against the fire blight disease that was present in the commercial pear trees, this tree was not compatible at that moment, means that it could not reproduce and it could not be invasive at that point of time. But afterwards, different cultivars of the trees were developed and they start cross pollinating with each other, and it made the tree invasive later on. So currently, this tree is present in 37 states and invasive in 13 states of the USA. So just a little bit about the plant pollinator interaction. This, as I said, like this tree was self-incompatible, and it used to be like it reproduced either by root sucker or cross pollination. So because of the presence of different like pollinator community and proximity of the trees cultivars to each other, they started cross pollinating, and that is why we can see the importance of pollinators in even the invasiveness of the plant species. So why is plant pollinator is that like important to the native species, or what is difference between the invasive plant and native plant, and why is it important? So answering that question, the effect of the pollinators on the native plant is actually conflicting. It is because some of the cases and some of the studies, it is found that the pollinators positively affect the native plant species by increasing the invasive plant positively affect the native plant species by increasing the foraging resources for the pollinators. However, in some other cases, because of the presence of the invasive plant, there is a lot of conspecific pollen transfer, due to which the flower cannot develop properly and produce nice fruit. So this is where they affect the native plant negatively. So moving on to the next slide, the problems with these trees is that it has a lot of problem. So first, it is competing with our native plant species. As you can see in the picture, there is like a lot of calorie pair growth in the our pine forest. So it is very difficult to control because of its long sharp thorns. It's like really hard to harvest these trees, and it has like very bad smelly flowers. It break easily in the wind, causing the trouble to the human beings and the road blockage, etc. And it has a very long seed dormancy, and also it is avoided by the arthropod herbivores, because of which they are like very less natural enemy of this tree present in the environment. So because of all those reasons, like different states in the USA has putting the ban on the commercial sale of the calorie pear, starting from Ohio in 2023, South Carolina in 2024, and there are other states adding to this list. So our main objective of our research was to identify the primary flower visitor of calorie pair and quantify the diversity matrix. Because this is the very preliminary stage of the research and no one has identified the pollinator associated with this tree. So our research and finding are very basic. So this is where we conducted our study on. We had total of 6 sites and we collected total of 706 pollinator species. So for the methods, we had like blue vein trap on the middle, and then there were like triangular, like 3 different cups of 3 different color on each corner of our field. So we set the trap out in the field in warm sunny days where the temperature was like above 50 degrees Fahrenheit. It is because the pollinators are active only during that temperature. So we set the traps at the sunrise, and the blue vein trap we collect every 12 hours. However, the cups we collect after every 24 hours. The in the blue vein traps, we were supposed to collect the insect live and then just capture the picture and set them free. But unfortunately, blue vein trap did not capture any pollinator for some reason, and the cup were like filled with the soapy water to reduce the surface tension and capture the insects. We also sweep netted the trees like 5 minutes per tree, and we selected 5 trees per site. So for the lab processing, we had to like wash all of the insects, like rinse it, dry it using the dryer, paper towel, all the method we possibly can. And then we pinned it and label it on the lab. And we had to further take the help of the expert and then different keys and internet resources to identify our pollinators and put the final labels on which family, order, genus that belongs to. So for the statistical analysis, we use RStudio. We did Pormanova pairwise comparison between different methods. We did the indicator species analysis, and we also identified the Hill numbers and collection method. Hill numbers indicate three different diversity indices. First is species richness, second is exponential of the Shannon index, and last one is Simpson index. So for the results, like we had a lot of like four pages of the result. I have just like included here our top results. So as we can see that the Toxomerus species in the Syrphid family was the major pollinator species for the calorie pair. So that was present in higher number, 189, and almost 25% of all the insect species that we collected. We do have other important pollinator species identified as the pollinator of the calorie pair. That includes Cyphus, Helophrydus, Andrina, and different other species of the bee family as well. So moving on to the Hill number, we can see that the yellow ball has the higher number of the Shannon index, but however the sweepnet has the higher number of the species richness, and the Simpsons and Shannon index both. And also the sweepnet is considered the best method because it directly captures the flower that were pollinators that were present in the flower. So it is considered as the better method over other methods for collecting the pollinators for our study purpose. So considering the fly side, we can see that one of our side had the higher species richness, higher Simpson index. However, as you can see, there is not like much difference between the sites in terms of the species diversity and species richness. So we can say like overall that all of our site has kind of equal diversity in terms of all of the indices that were used to measure it. So next, our ISA result shows that only like 4 species were related to a specific method of collection. That means that Toxomerus was related to yellow ball. It shows that the yellow ball method was efficient to collect this species, so that in further research in the similar species, other like future research can use this method. And also sweepnet method was able to collect like 3 unique species by itself. So the Pormanova analysis shows that the site was not like significantly different, but methods were significantly different from each other. So we can like it is always recommended to use the combination of different methods when you are doing the pollinator survey. So, and the pairwise comparison among the methods shows that the combination of different traps were better. So overall, like what we can see from this result, like overall that we can use the different combination of the methods when we are doing the study of the pollinators. However, because like our sites, like there is no difference, so other studies do not need to like consider more sites. But because like our site were very close to each other, they can do like far away in different states. It can be different, but not like this close. Like so. Overall conclusion, we can say that we collected like 756 individuals from 16 families and 3 different orders. And our result shows that the Syrphid family and the Toxomerus species from the Syrphid family was the major pollinator of calorie pair. And our study of the diversity shows that it is not significantly different among the sites, but it was different among the method. So this study just provide the baseline information on what major insects that are visiting the gallery pair, but there is a huge the future study, there is a like different kind of studies that other people can do based on our paper and our information that we have identified so far. So this study is very important to identify the pollinators of the calorie pair, and this will definitely help in the management of the calorie pair as well. So this is all for today. Thank you all for listening, and if you have questions and any comments, that are most welcome.
Thank you very much, Prabina. So I would encourage everyone at the end of a talk to click at the bottom on the little smiley face reaction button and join me to clap our speaker when they did a good talk. If you click on that button, you also see the raise hand. So if you have a question, it's where you can click so you can ask your question. Does anyone have a question for Prabina? So in the meantime, I do have a question for you. Prabina was wondering, you're saying that the tree smelled very, very bad, right?
Mm-hmm.
And that there were very few natural enemies or herbivores. And is there a reason for that? Because even if it smelled bad for us, usually when something has a smell, insects, there is always some insect that finds that attractive and delicious. And so is there a reason why this tree has very few natural enemy or herbivores?
So it's all about the nature enemy is just related to the leaves because the other species are not feeding on the leaf of this tree. But regarding the smell, like that is so bad for me that I have to change the dress every time I go out to the field. So, but like as we had already captured like so many pollinators, that means that the flowers are still attracting to those insect species.
Okay. And oh, we lost Prabina, but are you still with us?
Yes.
Ah, great. I think Todd has a question. Go ahead, Todd.
Thanks, Quentin, and thank you very much, Prabina, for a really interesting talk. So, a naive question for you. Honey that is produced from pollen, is it tainted? Because you said that the smell was so bad, and you and Quentin were just talking about that. So does it ruin honey?
Yeah, so because the tree is invasive here, this tree has not been used like that widely for the commercial purposes for the honey production. But we do see, like we did see it when we were doing the research in the suburban areas, like some of the bees, like honeybees that we collected were from actually the hives. So yeah, this is the interesting topic like that people can further explore and do the research on, like if this affect the quality of the honeybee, but so far we have not found any of those kind of data to like tell you for sure.
Okay, thanks.
Mhm.
Thank you, Todd. And I think we're going to move on to our next speaker. So thank you very much, Prabina. Give her a round of applause. And our next speaker is going to be Dr. Adriana. Oh, let me remove that. Dr. Adriana Corrales. Dr. Adriana Corrales is at University of Rosario in Colombia. She has a PhD in plant biology and a very, very strong interest in fungal community dynamics. She's a very prolific scientist with about 40 publications, 1,500 citations, and all of that include some very high impact factor journal like Trends in Ecology and Evolution. Adriana, I hope you are ready for your presentation. Sorry to tell you it was the previous one, but hope you kept the stress in you for now.
No, thank you so much. It's such an honor to be here and a privilege to be speaking to this audience. Can I share my screen now, right?
Yeah, if you can share your screen.
Okay. Sorry, I'm at the end of my presentation, but now
No. So here's my my original title that I have titled this talk Tropical Phagales as a Keystone Species for the Conservation of Tropical Ectomycorrhizal Fungi. But I saw and I thought that based on my audience, this was very fungi centric. So I decided, as a forester myself, I'm a forest engineer, that this should be more focused on showing the importance of mycorrhizal system for conservation of restoration of tropical trees. So that's what I'm going to be talking about. I also wanted to mention that I work for SPON, the Society for the Protection of Underground Networks, which is an international nonprofit organization, and I work in there as a scientist and as the lead of expeditions. So what is the importance of mycorrhizae? There are two major types of mycorrhizal fungi, arbuscular mycorrhizae and ectomycorrhizae. Arbuscular mycorrhizae are associated to more than 80% of all plants in the world. They specialize in phosphorus acquisition. There are a few species, we think it's about under 500 species globally. And they are very, very old because we think they help plants to colonize terrestrial environments. We also have the ectomycorrhizal species of ectomycorrhizal fungi that they help more in the acquisition of nitrogen. There are many, many species, about 25,000, but probably many, many more, and they evolved more recently. So what is the importance about this? The type of mycorrhizal association that is dominant in an ecosystem, in many cases, will determine many key ecosystem processes below ground. For example, nitrogen cycle, carbon cycle, phosphorus cycle could be influenced by the type of dominant mycorrhizae in the system. The tricky part is that mycorrhizae are not evenly distributed. These two types of mycorrhizae are not evenly distributed in the latitudinal gradient. So we have a dominance of ectomycorrhizal associations in the high latitudes, either in the north and in the south. And in the tropics, we have a dominance of arbuscular mycorrhizae that have very, very different nutrient cycles. I work with something that is rare, that is tropical ectomycorrhizae. So they are not super abundant, they are not the dominant kind, but they are there and they are very important. And that's what I've been trying to convince people about for a while now. So I wrote with my collaborators, Terry Henkel and Matt Smith, this Stanley review about tropical ectomycorrhizae, showing that there are main important groups in all tropical areas. So it's an interesting topic. So what we haven't demonstrated is that these mycorrhizae are important for propagation and restoration and conservation in the tropics. I'm going to show you a couple of, or like three examples of things that we are doing in Colombia with mycorrhizal fungi for tree propagations. So in this paper that was recently published in Nature Microbiology by Avery et al., they discuss the importance of defending the terrestrial microbiome, and they focus a lot on the importance of mycorrhizal fungi. They analyze a large data set of live in soil inoculum in tree propagation, and they found that live inoculum, live soil inoculum, help plants to grow 34% bigger than plants that were not inoculated with live soil. So I decided, well, before this, I was working in few experiments that I'm going to show, and a couple of them are from my students that I'm very proud of. This experiment was led by Mariana Carrillo. This is in collaboration with WCS. And we focus on Juglans neotropica. Juglans neotropica is not an ectomycorrhizal plant. However, it's a plant that belongs to a family that forms ectomycorrhizal and arbuscular mycorrhizal associations, and therefore we think it has very different root microbiome or mycobiome. It associates with a different type of fungi. So we decided to collaborate in this restoration project that was planting seedlings. And when we asked them, why don't we try to inoculate these seedlings with this live inoculum from other Juglans neotropica adults nearby? And we also compare your traditional just soil mix and see what happens. And what we found was that actually Juglans inoculum helps seedlings to grow more. So if you see here, I cannot find my cursor, my pointer, but if you see here in the left, the N treatment is the native soil treatment, and the V treatment is the vivero in Spanish. So just the greenhouse, the traditional mix that they used. And we did find significant differences with seedlings with the native soil growing bigger than the seedlings that weren't inoculated. Also, not all inoculum are the same. We tested 3 different juglans trees, and we found that one tree in particular was better as an inoculum than the other 2. We also found that the origin of the seed matters. So we were working in Amoja. Our inoculum was from Amoja, and we have seeds from Amoja. But we also brought seeds from Boyacá, another place. And we found that since local seeds from Amoja responded more strongly to the inoculation than seedlings from Boyacá. So seed origins matter. And this was in terms of high growth rate, but also leaflet production. So it was more responsive. And we found something really, really interesting as well. We took these seedlings and we sequenced all the fungi in the roots. And we found that seedlings that were grown in a sterile pit were actually showing a lot of pathogens in the roots. And this is a little bit concerning. We have no idea what is going on, but that pit left a legacy in all the seedlings that were grown in the greenhouse that were propagating using this substrate. So you see here the TE treatment is the sterile turban, and that blue line is the amount of pathogens that were in that substrate. If you compare it with the N or SN treatments, which are the native seedlings, the wild seedlings, they don't have that much of pathogenic fungi in the roots, but all the seedlings that were grown in the greenhouse had it. And this was confirmed when we analyzed the fungi communities. We saw that there were Fusarium and Dactylomectria that are potential pathogens. They were overabundant in these seedlings, while in the native seedlings, we have just soil saprotrophs. So that was an example of why this is important. It's important to think about an underground perspective. And this is another example based on Coccolobifera, the sea grape. This is a tropical ectomycorrhizal species. This was done in the north of Colombia with another of my students, Luisa Fernanda Chavarro from Universidad de los Andes. And with Luisa, we set up a very similar experiment, but we did it we compare growing seeds in peat versus growing seeds in native soil, and then transplanting those seedlings to commercial soil or native soil, and that makes for treatments based on the initial germination and then the substrate that we use to grow the seedlings. And we did find very similar trend. We found that for high relative growth rates and total biomass, mostly that seedlings that were germinated in native soil grew more than seedlings that were germinated in peat. So that was that we're finding a trend with that. And we also found that seedlings that were germinated in peat and transplanted to native soil didn't grow as much. So the earliest stages, the inoculum that seedlings are exposed at early stages matter a lot. So when we sequence the roots of this, and I'm showing here because there is a mix here of the sequences in the substrates and sequences in the roots of the seedlings. So the bars that have the seedlings are the ones that are no substrate. We found that seedlings in the wild have a lot of mycorrhizal fungi. You see is the orange bar, the orange part of the bar, and the seedlings that were grown in native soil and transplanted to native soil have a lot of ectomycorrhizal fungi compared with seedlings that were germinated in native soil and transplanted to commercial soil, or were grown in peat and transplanted to native soil. And it was curious because the seedlings that were germinated in peat and transplanted to commercial soil did not have any fungi in the roots, and we couldn't even amplify the PCR using fungi-specific primers, so they didn't have any mycorrhizae in their roots. So now, and these were examples to show you how important it is to take into account the underground perspective, and particularly the mycorrhizal fungi in tree propagation and tree restoration, and how that could affect the soil, the health of these trees. So now, talking about tropical oak forests. So these tropical forests are very, very interesting here in Colombia because this pie chart is showing the percentage of plants with ectomycorrhizal associations in all the Colombian Andes. And if you see, only 0.6% of all the species of plants present in Colombia form ectomycorrhizal associations, and those are the oak forests. So you can tell that oak forests are very different, are basically an outlier in the mycorrhizal world in Colombia. So I'm going to talk about this project I've been developing with some local collaborators, Dr. Andrés Abella from Universidad Distrital, and César Augusto Parra from Universidad de Rosario, Juan David Rosales, which is one of my undergrad students. And this is a project for in situ conservation of Trigonovalanus excelsa. In Colombia, Trigonovalanus excelsa, you probably haven't heard of this genus because it's a genus that is very rare. In the Americas, it's only found in very small populations in Colombia, and there are only 3 species. The other 2 species are in southern China. And this is a very endangered species of tree, and it's a gigantic tree. It's beautiful. The ecology of these forests are really special because they form tropical monodominant forests, and they do form ectomycorrhizal associations in a matrix, a background of many, many species forming arbuscular mycorrhizal associations. So they have very different fungi in the roots, and they accumulate an enormous above ground biomass. It has been shown that it could be even higher than some Amazonic forests. So this is a relic plant species. It's very, very old. It's believed it has arrived in Colombia in the Tertiary through the Panama Canal, or before, no, even before the Panama Canal, and the Isthmus, and is very, very interesting, very, very old, and is a mountain species. So very, very threatened by climate change and by habitat destruction. And this is the page from the IUCN. Well, this species, nobody has been able to propagate it in the past, and we were able to propagate it for the first time massively because we knew that special trick of the mycorrhizae. So nobody thought that the mycorrhizae would be necessary for these plants to really thrive. And since most of the species in our context are arbuscular mycorrhiza, arbuscular mycorrhiza, they were not inoculating the seedlings with the right inoculum. So unfortunately, we didn't do a control experimental design for this because we have a mass seeding event, and we just really had to plant everything, but we plant everything with very small inoculum from live soil, and they all survive. No, they all survive, but we have a very high rate of success with the reproduction, and all because of the trick of the ectomycorrhizal inoculation. So the fungi matter a lot for tree health and for restoration. We have found that these only hosts can be associated to over 200 species of ectomycorrhizal fungi, and many of these species are new to science. So if these plant species disappear, we're probably gonna lose a lot of fungal species that are unknown to science. And we also have found that different populations of the tree show different, associate to different mycorrhizal species. So for the conservation of the plant, we need the fungi, but for the conservation of the fungi, we also need the plant. So it's a dual beneficial idea, I guess. And I just hope I convince you that this is an important topic. And I don't know if I have more time or not. I'm doing on time.
If you want to wrap up in a minute or two, that would be great.
Okay, yes. Just the conclusion. I don't have a conclusion slide, but I just hope I convince you to, we cannot think about forest health and forest restoration only looking at the above ground processes and above ground biodiversity. We have a lot of important biodiversity that are below ground, and we need to take into account. I also wanted to mention here that since I work for SPON, we have a grant for exploring the mycorrhizal biodiversity in the world that is open. So if you want to apply, please contact me. I'm going to put the information in the chat, and please protect the underground. Thank you so much.
Thank you very much, Adriana. You convinced me that it's a very important work. Do we have any question for Adriana? I see a question in the Q&A from Kira. Hi, very cool presentation. Just curious why the oak uses ectomycorrhizae over arbuscular mycorrhizae.
Yes, that's a good question. It would depend on the species. It would really depend. And then it's an important functional trait for the plant, and that we really need to take into account. So when you are working with a species, just learn about the background, learn about the type of associations it has to help your restoration and your propagation efforts.
I see we have more question in the chat box from Juliana. Adriana, congratulations. Also a scientist from Colombia, and your work on the hiding forest is amazing. Thank you for sharing it with us. I guess it was not a question, but
thank you so much.
Word of appreciation. Okay, we're gonna move on to the next speaker. I just want to emphasize, I forgot to emphasize with our next speaker, but we ask our speaker at the end of their talk to move to a break room. So if someone from the audience want to go a little bit deeper with our speaker or a bit more interested in their work, they can talk with them a bit more deeply in private. So please don't be shy to go and chat with our speaker. In the meantime, I'm going to move on to our next speaker. So unfortunately, Dr. Nari Williams couldn't be with us today. So our next speaker is Dr. Kamal Gandhi. Dr. Kamal Gandhi is at University of Georgia and has her own lab there as a Gandhi Forest Entomology Laboratory. Research focus on forest insect disturbance, chemical ecology, and invasion dynamic. She has about 30 graduate students and postdoc, more than 160 scientific papers, and almost 3,000 citations. So we are very happy to have you with us, and we are looking forward for your presentation, Kamal.
Well, thank you so much. It's such an honor to be here today, and thank you for your kind invitation to come and talk about P4 and how it links to the needle diseases. So I'll share my screen. Right. Let's see. I make it big. Is this
Perfect.
good?
Yeah, all good.
Okay, awesome. Good morning, good afternoon, wherever it is in your part of the world. I'm thrilled to talk about an initiative we've been working on for the last, I want to say 3 to 4 years, and it's really gaining steam now with time. It's called the Pine Pandemic Preparedness Plan, or the P4, and the gist is to really try to proactively protect our southern U.S. pine forests. And I have a collaborator on the project, Dr. Kia Klepsik, who's with the Jones Center at Itchway. So let's suppose we go for a walk in the woods, and it's a beautiful day. We're walking around, things seem to be looking really good, and suddenly we start noticing crown dieback, symptoms that we have not seen on pine trees before. So as we are scratching our heads, we dig a little bit deeper, we find maybe these purple insects or these actually really beautiful looking mushrooms, but any kind of insect or pathogen that could be impacting our pine trees. And for those of us who are in the southeastern US, we know that pine trees are very important, both ecologically and economically, and it does stop us in our tracks and to pay more attention to what's happening in our forests, both at the stand and at the landscape levels. So what do we do now? Are we really prepared for the next high impact pest or pathogen that could be impacting our pine species? What about our neighbors? Considering 80% of the land, for example, in Georgia is forested and about 80% of it is in private hands. What about the company? If you're working with a private company, or if you're working for state or federal agency or your extension agents, what about us scientists as a community? Are we truly prepared for a pine pandemic to start operating in our stands and in our landscape level? I mentioned southern US pines are extremely important, both ecologically and economically. They also happen to be extremely vulnerable to high impact species just because we have a lot of high number of ports of entries. They are grown in monocultures over vast landscapes and at the regional level. And a lot of different genotypes of particularly one species, loblolly pine, has been planted in many different places. And those pine plantations actually continue to grow with time with intensive silvicultural treatments. And it could have all sorts of cascading impacts from to regional economy to many different ecosystem services that could be disrupted in these stands. So there's really has been a need for a proactive approach. Most of the approaches we have done to high impact species in the past have been reactive. And as time goes by, we are paying more and more attention to this so that we're not sitting ducks and we are better prepared in case of a pine pandemic. So it really started with discussions. Let me see. In the last few years, and basically Kira and I got together actually during the COVID-19 pandemic and start scratching our heads. If we could put together a large team of folks, which we did over here, what we called as the core committee members. These were what we call as thought leaders. They're highly respected in our community. There's big picture thinkers. They're in diverse agencies, as you can see over here. And the core committee members pull together what we call as a task force members. And these are diverse folks in different agencies, but also in different disciplines. So they not only represent forest entomologists and pathologists, but they also represent soil scientists, modelers, silviculturalists, and so forth. And so we've been in meetings almost every few months, and we came together with what we call as the phase 1 of the pine pandemic preparedness plan on recommendations on how to deal with the next high impact species in our forests. So it's really been a collaborative grassroots effort that came up from the bottom up. There was no bottom down. And I also want to mention at this point that we actually had no resources when we started working on it. We actually didn't get resources until the last year. The needle disease issue came up. And it was really run on the premise that we want to get together and talk about invasive species for the greater good of our forests. So we have this working model. So here we have all the different agencies on top. You can probably add a few more agencies to that. All these arrows represent a lot of communication and back and forth happening between the agencies, but also some of the initiatives that we brought up in these rectangles at the bottom. The main thing, take-home message is that there is a central hub that we recommended that will collect, synthesize, and also widely disseminate information between all these different entities, but also from the different parties working from whether it is early detection or diagnosis or delimitation or coordinating efforts in terms of response teams. And the central hub is round by intent because it meant to move back and forth between all of these entities and different parts of trying to respond very proactively and quickly to a potential pine pandemic. So we were brought in by the request of several major entities in our region about using this P4 model to the existing pine needle blight outbreak. And to give you a quick background about that, it's really centered around the brown spot needle blight, which is caused by this fungus. And it was reported, it's been reported on pines for a very long time, and it's considered native to our region. But it was first reported in South Carolina, but it really hasn't caused problems in terms of dieback or mortality of pine trees, particularly loblolly pine trees, which are the backbone of our forest economy in the region until in the last, I want to say, decade or so. And so because it's mostly emerging as an issue in planted pines, which are economically valuable, there has been a big focus and a big push on trying to understand the ins and outs of the brown spot needle blight and to do a very cohesive and concerted efforts towards it. So basically, we've had a bunch of meetings so far. We have another meeting coming up in March. We're going to have an in-person meeting, but there are some issues with travel restrictions. So we decided that we're going to look at the 4 phases of the P4, which I'll describe in a second, and then use this framework of the P4 and put it towards the needle disease issue to really address it in a cohesive manner. And in my mind, I go, we should have a 1-year, 2-year, and a 3-year plan. We should not have a 5-year plan because by the 5th year, we should have resolved this issue. And really, the gist is to look to see what is being done, what needs to be done, and how we can work together effectively and use our limited resources to make a heavy impact towards this needle disease issue. So I mentioned the 4 phases. Here they are: communication, detection and diagnosis, delimitation and assessment, and response. So I want to quickly go through those and hit some few points as they relate to the needle disease issue. So the first one is communication, which underlies everything, all parts of the P4 in reality. And this network, this communication network, is absolutely critical. Good news is we already actually have great networks in existence, and they provide us a strong foundation, but we need to build on those networks and create new networks needed as well. And they have to be from all the different levels so that we are cross-communicating with each other in real time. And because of that, we hope that any kind of changes, deviation from the norm in terms of tree mortality would be reported immediately. And for that, we are talking about region-wide system for evaluating the tree damage and coming up with very standardized protocols, for example, so that we can actually add statistical power to what we are seeing out in the woods, or other folks who are in the woods a lot more, add a lot more credibility to those observational data. And one of the outcomes that has come out of the communication is so much is already being done. And that was one of the reasons why we got together, is who's doing what, so that we're not duplicating efforts. And then looking at the big picture, we can start queuing in different parts as needed based on our expertise, because everybody has something different to bring to the table, which is equally important. But perhaps there are ways to coordinate our efforts a lot more effectively. So these are just some of the examples. This is not all the things that are being done on this needle disease issue. So second phase, the detection and diagnosis, which is absolutely critical if you want to do early detection and early intervention. And I think this depart from expectation is one of the thorny ones that we have run into. How do you figure out something is deviation from the normal mortality or the background tree mortality levels? And actually, a lot of times people have been reporting mortality in other instances, and they don't get picked up, or we don't make sense out of it really quickly. And by the time we pick up that something is seriously wrong, it's far too gone. And part of the detection is also figuring out like what's different from the native causes. And we're actually working with the Forest Service very closely. We're in the process of hiring a postdoc who will actually start tackling this particular depart from expectations much more in detail. Diagnostic component is extremely important, because if we don't know which species we are dealing with, well, we may end up with some very different management recommendations. For the brown spot needle blight, we know which pathogen it is. Having said that, we also know that they are isolated. The incidence rates are different based on where you are in the states even. And we also wonder if they're actually a complex of needle pathogens that are operating in these stands. And as you notice from this loblolly pine tree, a lot of dieback is from the bottom up, which I think is typical for the needle diseases. And for the diagnostic expertise, we have to rely on other folks. So we want to build these diagnostic networks right on the start, so that we are not trying to figure out who to send the samples to. We already know who to send the samples to, and the information is coming back to us in a very quick and real-time manner. The third one is delimitation and assessment. This is a map from information given by many, many different folks across the region. As you can see, this is for the last 7 years or something. You can see some of the positive reports coming out of Mississippi and then expanding with time. We don't know if the needle disease issue had already been causing issues for a while and we're just picking it up more, or we are seeing greater incidence of this with time. And you notice that these are all at the county level, which is a very coarse level, but we really want to get down to absolute pinpoint levels where they're operating, so that we could create better detection tools for the adjacent areas around it, and put them on a watch list, so to speak. But it does seem that we are more and more reports of brown spot needle blight is coming up and impacting trees in our region. So it is a regional concern to a large extent. And because if we have those pinpoint spatial data, we can also ask different questions related to genetics. That's a big one. We do think there's a genetic basis to it. So a stand would be impacted, but the stand next to it won't be impacted. Well, what are some of the reasons why that could be occurring? We think genetics is one of it, but it may not be the only factor out there at the landscape level. And it also asks us to prompts us to ask a lot of different questions. What has changed? It has been reported a lot on longleaf pine before, but it has never been a problem. Has it become more virulent? Are there different varieties of the brown spot needle blight? Climate change is a big one. Has something changed with the climatic conditions, and especially with the climatic conditions and the interaction between the loblolly pine genotypes? There are literally hundreds of genotypes being planted each year in our forest. So what are these interactions happening, and which will translate back into the management focus? And the last one is a response. And the response is a big one. And this was the one that we actually had the hardest thing with the P4, because how do you provide recommendations on what to do if you don't even know which species it is? But in this case, we think we know the main player. There could be a few more players that are out there. And in reality, our stakeholders, the foresters, they're interested in knowing which species it is, but what they're really interested in knowing is what are we going to do about it and how are we going to manage it. And it will require a lot of cooperation, just like everything else. But this one especially require a lot of cooperation because we can run these large-scale experiments in terms of different management interventions and do it in a standardized way to see if they actually make a difference. And we may have to use many different kinds of management activities. I mentioned silvicultural, but maybe fungicide, maybe prescribed burning. And in some cases, some of the information is already known from other pathosystems that we can bring in. So none of this is going to be done in isolation. It has to be very deeply grounded in what is known about a particular pest or pathogen in the other countries and what we can do about it, potentially do about it here in the southern US. And some of these partnerships are already occurring across the region, which is a very heartening thing. And there are a lot of great scientists working on tackling this problem and doing different parts of the responses. So these are just some of the examples. I don't want to go through all of them. Having this actual screening center is really great. They set up a fusiform rust, and they are working on setting it up for the needle disease issue. Obviously, detection and so forth follows all in it. Prediction is going to be a big one. Mentioned silvicultural approaches. And then if there is, which it seems to be a strong genetic basis to it, we may want to deploy resistant host material, particularly in areas that are pretty high hit. And perhaps even bring fire back into these systems, because in longleaf pine, it's not really a problem, primarily because they're burned every 2 to 3 years. So fire keeps the spore inoculum really down in these stands. So to conclude, when we started working on the P4, we weren't dealing with the needle disease issue. And we were really focused on non-native species, to be honest. And this one seems to be a native species that is behaving differently and is causing significant economic impacts. So something is happening, and it's becoming to be a huge issue. So this is the time to act. It will require a lot of talking. I think that's what I do all day, is talk all day. A lot of coordination, and there's a lot of push within and outside the community to share as much information as possible so that we can all collectively move forward and really resolve this issue. Like I said, in 3 years, we should be done. Let's see if it's a moonshot. I would like to really hit that moonshot if possible. And if you have any questions, both my email address and Kier's email address is here, and we'll be happy to answer any questions related to the P4, but also about this needle disease, larger needle disease working group effort that we have. Thank you very much.
Perfect, Kamal. Thank you very much for sharing with us today about your work. Do we have any questions for Kamal? I see a lot of clap for your talk, so happy to see that. I don't see a raised hand, but I do have some questions for you while people think about it. I see Prabina has a raised hand. Here we go. Go ahead, Prabina.
Hi, Dr. Gandhi. It was a really great presentation. But I was wondering, like in the loblolly pine, like we have seen loblolly pine going through a lot of diseases. Like sometimes they are affected by apes, sometimes they are beetles disease, root rot disease, and now this is a new disease. So do you think that we have to go like a holistic approach to like deal with all of the different disease because of the genetic or some reason together, so that they don't have to face any new disease in the future? Or like how do you think like we can deal with that so that like there is no like because like they have, I feel really bad for them because they have to deal with a lot in the southeastern southeastern US. So
Yeah, well, thank you for the question. So I wish there was one or two silver bullets that could resolve in all the forest health issues we have related to loblolly pine. But there's another way to think about it. Loblolly pine is an early successional species. The reason why it's chosen is it grows relatively fast, right? That's what we would expect early successional species to do that, as compared to longleaf that grows slowly. So I like to think of them as opposite end almost of the resistance spectrum. And resistance is never a two-factor model. It is a scale. And I think loblolly is on the other side than longleaf. So if we're going to put a pine species out there that is selected for growth and other stem forms, it's probably not going to allocate as much resources into defense. And it's the growth versus defense paradigm, right? It's the trade-offs. And so, we have talked about it. Like, for example, can we screen loblolly pine for fusiform and the needle disease issue at the same time? In those cases, both of those we think has a genetic basis. Fusiform, I'm pretty sure we do. For the needle disease, we think it has a genetic basis. Maybe you can, maybe you can't. But a lot of other species like ipspark beetles, we don't have any evidence that there's a genetic basis for it, which actually doesn't make sense which makes sense because it's a secondary species. So the tree has to be stressed for some reason for the bark beetles to come in. And so, I think, and we also have to remember that we are doing these large scale experiments, almost regional wide experiments, where we're selecting different genotypes of loblolly pines for certain attributes and putting them out there. We have very little, well, now we're learning more information on how these genotypes are interacting with different native insect and disease species.
Yes.
So it's not surprising we're seeing this. It's not surprising that loblolly pine has a lot of issues. It is surprising that this native pathogen is apparently behaving differently.
Yeah, absolutely. Thank you.
Do we have more question? I don't see any, but I do have one for you because you talked a lot about early detection and in your presentation, and I know how difficult it is already for insects. So for pathogen, it's even worse. And usually, when you see it, you are already way too late, especially for pathogen. So where do you see the future of early detection? Do you see any kind of technology in the next generation of scientists or something that can help with that?
AI. We gotta go machine learning. Yeah.
Yeah.
Well, I'm kind of half joking, but not really. That is a big one because. So okay, let me just step back. The more eyes we have out in the woods, the more passes we are making through these stands on the ground, I think the better off we're going to be. And most forest companies will make, during the lifespan of a loblolly pine stand or even slash pine stand, they'll do 2 passes, 3 passes. Even that is enough. That should tell us what's happening in these stands. So now, about, like I said, 80%, especially in our state, is in private hands. If that information could be filtered back into a common database where, a lot of that information is protected in the sense the lats and longs don't go anywhere, but it filters out to a few select folks who are keeping an eye on what's happening at a larger spatial scale. That will be informative. But that's not all, because we get a lot of reports all the time, right? Things are happening all the time. So which reports are viable and which reports are not viable? And there's going to be a lot of noise in the data. So how do we filter out that noise? And I think that's when machine learning comes in. Okay. And sure, you can do aerial work, you can do satellite work, you can do remote sensing, you can do all of that. With something like the needle disease, the issue is the dieback is from the bottom up. And that makes it harder to detect it from really high resolution. So I think a combination of those, communicating very closely with the private folks and building those relationships, those partnerships, and the trust in the system, and then trying to filter out the noise in a time-sensitive manner is going to be the key. That's my take on it.
Yeah. Thank you so much. I think we had a few more questions for you, but we're gonna move to the next speaker. So I invite you, Kamal, and people who want to talk with you to move to the break room so they can ask a few more questions. And we're going to move on to our next speaker. So our next speaker is Dr. Beccy Ganley. Dr. Ganley is a plant pathologist and the leader of the innovative plant pathology group at the plant and food research in New Zealand. She did a lot of work on foliar disease and fungal on the fight and pathogen. Published about 20 articles, including some in prestigious journals like PNAS. Beccy, if you are ready, the floor is yours.
Yeah. Oh, hang on, just let me change that setting.
Huh?
Morena, good morning from New Zealand. It's 10 o'clock in the morning here. I'd just like to first off say thank you very much to Josephine and all the team for organising this. I think it's fantastic. So a big round of applause from me to them for doing this, and thank you very much for inviting me. So I'm going to talk about unwanted pathogenic invaders of New Zealand's forests. I sort of realised that most of the time when we head overseas, we talk about our exotic forests, but we don't really talk a lot about our native ones. So to start off though, just a few New Zealand facts. 49% of New Zealand is covered in native forest or vegetation. We have about 8% in exotic trees or exotic forests, sorry. And of that 8%, 90% is Pinus radiata. So it's a very strong Pinus radiata monoculture. But I'm not going to be talking a lot about Pinus radiata today. Some other facts: we're the first country in the world to give women the right to vote in 1893. We also had the first female head of government in the world to give birth while working. So we've got some really good statistics on equality for women, but we also have some less than impressive ones. So while I'm really pleased to say that we have equality in women graduating with science degrees, unfortunately 25% of our professors are female and 18% of foresters are female. So we've got a long way to go to get it throughout the whole sort of pipeline of employment. But we're getting there slowly but surely. So they were my facts. Now on to my actual presentation. So for New Zealand, we have a really, really strong focus on pre-border. We don't want things in. And we know that if you do get it in, you have a huge cleanup. I do realise that picture of the lady cleaning up is not necessarily the right picture to be putting on a woman in science. But at the same time, if only 18% of our foresters are women, maybe we do need to have a bit more balance in there. And if we had a 50-50, maybe we'd have some more success in our post-border cleanups. So pre-border is a huge part for New Zealand. We have some of the strictest biosecurity rules in New Zealand, in the world, and really limited on some of the things that we can bring in and out. It's a very strong system. So what does that mean for our native forest? So this is an example of our native forest. That's a beautiful Cody tree in the centre there, and I'll be talking about some of the diseases of that later. And this is what the predominant part of my talk is going to be on, is our native forest. As I said, I don't really talk about it much and realise that most people probably have no idea what things are really impacting native trees in New Zealand, but would probably have a much better idea of what impacts our Pinus radiata. So I'm going to start off with the exotic pathogens impacting our trees. So the first one is Ostropyxina sidii, which is causes myrtle rust in Myrtaceae species. So this is had a pandemic strain that's gone around the world. We've got over 480 species of myrtles that are impacted by this rust, which is really unusual because most rusts only do one or two. It affects at all stages of new material, foliage, stem, flowers and fruits. And you can see in the picture there some flowers that are affected, some capsules and some new tips as well. First detected in New Zealand in 2017, blew over from Australia. It infects at least 12 native myrtles. We're already facing localised extinction, which is following the same trajectory as what happened in Australia when they got myrtle rust. And unfortunately, infection and disease severity is increasing every year. It's for our most susceptible myrtles, it's a very poor outlook and there's not a lot of management options. So this is a really big one for us in our forests, myrtle rust. Another one is Phytophthora cinnamomi. It has a huge host range. Many of you will be very familiar with this. Over 2,509 hosts, causes root rot and dieback. It is present in New Zealand. It doesn't cause huge issues in our forests, as in we don't have trees that are dying from this, but it is found in conjunction with Phytophthora agathodicida, which I'll talk about soon, in our kauri, that's that tree at the start, Agathis australis. It is a major horticultural problem for us, and it does mean that the pathogen kind of flows across land uses. And I know that most of you should have hopefully heard of this one because it is listed in the 100 of the world's worst invasive alien species. So as I said, it doesn't cause huge problems, but it is a problem in our forests. That is the end of the exotic pathogens that are really having an impact in New Zealand's forests. And I've got to say, I'm pretty proud that I could only come up with 2. And even Phytophthora cinnamomi, it isn't killing trees at this point that we're aware of. It doesn't mean to say that we don't have others. We have this compendium of fungi, and it also includes oomycetes on trees and shrubs in New Zealand, which is exotic and native. And that has lots and lots of different things, but they tend to be very sporadic in isolation and don't necessarily kill a tree, or if it is, it's a one-off. So we do have a good knowledge of what's out there, but we don't have very many things that really hits that threshold of something that you'd consider to be highly problematic that you need to potentially actively manage. My next category, though, is that one of not really sure if they're native or exotic pathogens, which for me is that part of why it's so important to understand why we have, because I think that this is a problem. It has implications for management if you don't know if it's native or exotic. And it is something that worldwide we struggle with many pathogens. The first one is Phytophthora agathodicida. I don't know if we'll ever know whether this is native or exotic. It's been present in New Zealand since the 1970s when it was first discovered, but it wasn't until 2006 that it was really realised the devastation that it was having. So it causes a disease known as kauri dieback, and you can see in that picture, causes cankers and resinous bleeding, infects kauri of all ages and sizes. They can die. The pathogen is broadly across the native range of kauri in New Zealand. This is the only pathogen in New Zealand that has a national pest management plan. So it is a severe disease of a native tree. The kauri is only, its distribution is only at the very, very top, or the top part of the North Island, so it's not across the whole of New Zealand. And it does cause huge impact. So this is a big one for us. And as I mentioned previously, we have Phytophthora cinnamomi, which is often found in association. At this point, I'd just like to I've got two Phytophthora pathogens, and in some ways, just a little bit of a count, because that's relevant at the end, of how many Phytophthora pathogens that I talk about. So the next one is probably grasping at a long straw of a native pathogen impacting our forest, but it's quite a fascinating one. It's Phytophthora podocarpii. It's technically listed as being native to New Zealand. I'm not entirely sure that there's been enough work done on it to be conclusive of that. It causes needle blight in a species Podocarpus totara for us. It's been only recorded in our North Island, causes a needle cast and stem infections. It is sporadic. However, it's one of those ones that I put on my lists because I kind of think we've had a bit of a change in its expression. It's not something that we've seen previously, and it's unknown whether under changes in climate, whether we're going to see further expression of this. So it's probably one I'd list as a watching brief, but I wouldn't say that it's causing huge problems. So at this point, we're at our third Phytophthora. That's my end of exotic or could be exotic pathogens. And now I go on to so I've got 4, and some of them are a bit of a long straw. I'm on to my native pathogens. Phytophthora canovii is probably known to many of you, causes a lot of problems overseas. It's known to be a native species in New Zealand. It doesn't actually cause any known disease in any of our native plants. I did put it in there just because it's a native pathogen. It causes problems in exotic tree species. It was first detected in custard apple trees, and it also affects Pinus radiata. Its distribution in New Zealand is not really very well understood at all. Do know some areas where it is, but its range is poorly understood. So I've put that in there, acknowledging that it is a pathogen in our native forests, but it's not a pathogen that causes problems on our native plants. So that's 4 Phytophthora. And then just because I felt like I should include something with Pinus radiata pathogens, I thought I'd just give the list of what are our top ones for our, for one species that's commercially planted. We've got Dothostroma septosporum, Phytophthora pluvialis, Phytophthora canovii, Cycloneusma minus, Armillaria novae New Zealande, and Neonatria fuciliana. So already I've got more pathogens on this one exotic species than what I was able to list at the same degree, and something that's causing serious impacts and needs to have some form of active management around it. So quite an imbalance considering how many species we have in our New Zealand forests. So my final part of some of it is, what are we worried about? And that's our overseas disease risks. And this is part of what forms our biosecurity system. And for us, it's really important that we keep these things out. The first one on our list is rapid hair death. That's caused by Ceratocystis luku-ohia and Ceratocystis julio-ohia. In Hawaii, it's called widespread death of the Metrosideros species, Metrosideros polymorpha. In New Zealand, we have some close relatives we've got, I think it's 12 Metrosideros species. We have some close relatives of those species. And so there is high concern that if either or both of these pathogens came to New Zealand, that our native Mutaceae, Metrosideros or other genre could be impacted. And there's also concern of what it might do to some of our exotic species. So this one is high on our list of unwanted. Another one that many of you may be familiar with, Zayella fastidiosa. I actually went to, I talked to some Indigenous people in New Zealand about this one, and I always remember that one of the ladies said it's like a Harry Potter spell, and I always remember that. She's like, Zayella fastidiosa. So if you're ever struggling to remember it, just think of Harry Potter. It has a wide range. It is a bacteria. It causes a sudden decline, transmitted by xylem fat-feeding insects. We don't have sharpshooters in New Zealand, but we have several spittle bugs. We don't really have a good understanding of whether our native species will be susceptible. Obviously, some of the exotics, we know that they will be because of what has happened overseas, but we don't have a lot of knowledge. But because it has a wide host range, there is concern about this one to our native species. And it's certainly something from a commercial perspective that we absolutely want to keep out of the country. The next one is probably a no-brainer for people as well, Phytophthora morum, sudden oak death. Over 100 known hosts present in many countries worldwide, spread through the global nursery trade, dieback and bleeding cankers. And again, we don't really have a good understanding of whether our native species will be susceptible to this or not. So it is definitely one that we're not wanting in the country. So that's actually pretty much it from me for today. I just wanted to sort of recap on a few things, and that is, in short, in New Zealand, we don't actually have a lot of pathogens that impact our native forests. Really struggle to build up a list of things that are deeply concerning. And we have a couple of things that are sort of what I would list as watching briefs, of course, some minor damage that sit in there, but it is actually quite difficult. Having said that, myrtle rust and Cody dieback are two very, very devastating diseases, and they've only really emerged for Cody in the 2000s, and then obviously for myrtle rust it's been recently. So prior to 2000, I would have struggled to have told you of any diseases of our forests that caused huge concerns. I did mention to you to keep focus on the phytophthora that I went through. I actually have lost count of how many it was, but there are a lot of phytophthora species. They would be the group of fungal pathogens that we'd be most worried about. And I'm not sure whether that's something that other countries share. I'd actually be quite interested to know, where they rank in the same way in other people's native forests. Are they the ones that you're most worried about, or is it something else? But for us, there's certainly a lot of phytophthora that are causing issues or are of concern to us. It's not just down to, my last point is about the biosecurity systems. The lack of pathogens impacting our native forests isn't just because of a biosecurity system that's effective, but it has a huge part to play in it. We have been very effective at keeping a lot of things out of our country. We are geographically very isolated, so we do have an advantage over other places, but it does show you how effective a biosecurity system can be. And that's what I wanted just to touch on today is that part. So anyway, I hope you've enjoyed my talk. I'm happy to answer any questions, happy to follow up on anything. If you go back into account of your phytophoras and what things you're most concerned about, I'd love to hear. And welcome to get hold of me at any time. So again, thank you very much. And thank you to the organisers for this as well.
Quentin, you're muted.
Thank you, Josephine. Thank you, Beccy, for that presentation. I was busy not thanking you for running Harry Potter for all of us, but we do have a question for you from the chat box from Blitze Stone. What are some things you do in New Zealand that limit the introduction of harmful fungal pathogens?
It's pretty intense. So we have strict lists of what can and can't be brought into the country. This is an example for Pinus radiata. We've had one lot of germplasm that has been imported into the country since 1989, I think it was. So for a country that specialises in it, it can often be full restrictions on bringing germplasm in, germplasm being seed and live plants. Some species we can bring seed in, but we can't bring live plants. Sometimes you can bring seed and live plants. When they get to the country, they go into PEQ, post-entry quarantine, and they'll sit there for however many months, and they'll be surveyed and tested to make sure that they don't carry any harmful pathogens or insects as well, and they have to sit there for a certain amount of time before they are actually granted access to come through into the country. So from a commercial perspective, yes, it can be quite limiting, but from protection of our trees, it's an effective system.
Thank you. I think we have a question from Todd.
Hello, Beccy. It's nice to hear from you. And thank you for your presentation. So one of the things that I think is really interesting with respect to Kauri dieback and myrtle rust is that those trees are, culturally significant to a lot of different people. And you didn't really talk about how stakeholders have come together to sort of identify these different disease issues and then look towards solutions. Can you just spend like 30 seconds talking about what's happened? I think it's really neat. So
Yeah. No, I can. It is a really it's been a fantastic journey with some of these diseases. So historically, New Zealand was colonised around the 1800s. Been a huge suppression of the indigenous people. These trees are of immense cultural value to indigenous people. Kauri were the sort of father of the forest. Pohutukawa were involved in some of the processing of people when they died, and also they tied their canoes to Pohutukawa when they first arrived in New Zealand. So, and they can remember the trees that they tied the canoes to, they're still alive. So huge impacts culturally for them, and it's actually been really, really heartening. New Zealand has had quite a shift in how we do our science, and so now we are seeing Indigenous people being included into our research programs, and we're seeing it's not equitable yet, but we're starting to see Indigenous research being given a equivalent priority as what I would term a Western-based research, acknowledging that most of our colonisation was British, and we're starting to see this new knowledge system come in, and it's a completely different knowledge system and a way of doing science, but hugely evolved methods that they traditionally would use, and so the outcomes have been far greater than what one system could have achieved alone. And it's how we managed keeping the integrity of both systems, which is really can be quite challenging. And it has been quite a journey and a mind shift to get over a different methodology of science using a very different system.
Yeah, thanks.
All right. So we are running a little bit out of time. So I see Pascal had a question and Simon had a question in the chat box for you, Beccy. So I encourage you to go to the break room to ask your question if you can, or Beccy, if you can also later on answer the question in the chat box. Also, we had some comments that you can comment on. We would really appreciate that. Thank you very much, Beccy.
Okay, thank you.
Okay, so our next speaker and last is Dr. Deepa Pureswaran. Dr. Pureswaran worked at the Natural Resources of Canada at the Atlantic Forest Center in Fredericton, in Canada. Our research focuses a lot on forest insect pests, including their semiochemistry, host and mate finding behavior, community, and population ecology. Deepa has about 80 publications with more than 2,000 citations, so we are very looking forward for our last speaker of the day. Thank you, Deepa.
Thank you, Quentin, and thank you, Josephine, for inviting me to this group of distinguished speakers. I'm very honored to be part of this. I'm going to share my screen and you should see my screen.
So we see your screen. It's not in full.
Yeah.
Yeah.
Now it should be in presenter mode.
Perfect.
Is it good?
Yeah.
Okay, but that's not my first slide for some reason. There we go. So I was wondering what to talk about for this talk, and I decided to take you through my career trajectory and focus on a few highlights along the way. So to start out with, I was born and raised in southern India in the tropics, and I did my first degree in zoology in India, and then got a fellowship to work with John Borden at Simon Fraser University, which is near Vancouver in coastal BC. So I made the trip all the way, first time on a plane, and I was picked up by one of his students. And a few days later, John Borden took me in a pickup truck to a drive to the woods. Looked out of the truck window, and this is what I saw. And I had heard a lot about the fall colors, and so I admired the beautiful fall colors before I realized that the trees I was looking at were conifers. John shook his head, and then I realized that I was going to embark on a long journey studying the intricacies of communication in bark beetles. So I started off for my master's and PhD asking different types of questions on bark beetle ecology and behavior. A lot of them were pretty basic, like how do bark beetles select the right species of host? So each species of bark beetle attacks a different species of conifer. So pretty much every species of conifer has its own specialist bark beetle. So other questions were, how do these conifers differ in their chemical profiles? What do these beetles use to detect these conifers and choose the right hosts? And since they have this phenomenon of mass attack where thousands of beetles aggregate on a tree to colonize and kill it, there were questions about how much pheromone do these beetles produce? Do larger beetles produce more pheromone? So these were some of the main questions I started asking. Over the course of several years, we found the answers to some of these questions. So we found that beetles differ in their aggressive potential to overcome a tree's defenses and kill it. And in the more aggressive tree-killing species like the mountain pine beetle, beetles actually land randomly on trees and test them, followed by a close-range olfactory or gustatory rejection of non-host. So non-host trees are rejected after beetles land on them and test them for suitability. We also found that the different conifers were similar in their actual qualitative profiles, but the relative amount of the different terpenes that they possessed varied. And it was this quantitative variation that beetles actually could use to choose the right species of host. With respect to size and pheromone production by individual beetles, we found that there was no relationship between size and how much pheromone a beetle produced, even though individual variation among beetles was high. And this was what took me out of what I thought was the cold West Coast to even colder climates. So when I finished my PhD, I was intrigued by the result that we got. We found that a lot of beetles produced very little pheromone, and a few beetles produced a lot of pheromone. And this was true not only for the species that I was studying, but it was true for other species as well, like the southern pine beetle and the European spruce bark beetle. So pheromone production is very critical to bark beetles because they have to attract a threshold of number of beetles in order to colonize the tree. If not, the tree's defenses can potentially kill them. So beetles essentially either survive as a group or they all die as a group. So I wrote this paper that was inspired by one of the professors with whom I took a behavioral ecology course that essentially said that there's this phenomenon of group selection where natural selection acts at the level of the group that allow low pheromone producers to survive and reproduce if they were part of a big group. And I submitted this paper to, I think it was Oikos, and it got rejected. And I submitted it again to another journal, and it got rejected. It got rejected so many times. And the last time that it was rejected, the reviewer actually signed his name, and it happened to be Matt Ayers, whom some of you might know, out in New Hampshire. So he just didn't think my argument was solid and that group selection did not exist in bark beetles. So at the time, I didn't have very much to do. I had finished my PhD, and I got an NSERC fellowship to do my postdoc somewhere. So I wrote to him and said, well, how about I come over to your lab and let's figure this out. Why do some beetles produce so little pheromone and can still survive? So I made the trip across the continent to New Hampshire, only to find that the lab at the college, Dartmouth College, was locked because everybody was down south in Mississippi. They were studying the southern pine beetle. And so I flew to New Orleans, drove to Mississippi, totally different environment from BC, where the southern pine beetle was active. So it's interesting about the southern pine beetle is this was almost 18 years ago when I went down there. All the outbreaks were down here in Louisiana and Mississippi and Texas. And now the beetle populations have been detected as far north as Maine. So a lot has changed over the years. So anyway, when I was down there, we asked questions like, why is there individual variation in pheromone production? Do beetles that arrive early in the attack sequence produce more pheromone? Because there's a disproportionate variation in pheromone production relative to how important this trait is. Do early arrivals have more offspring than the late arrivals? So is there a correlation between high pheromone production and fitness? So over the course of the next year and a half or so, we found that actually most beetles arrive in the middle of the attack sequence. And beetles that arrive in the middle of the attack sequence are more fit than beetles that arrive either too early or too late. So there was stabilizing selection that favors beetles that attack in the middle of the sequence. It's not a good idea to go in too early or too late on the tree. So this actually favors temporal synchrony, or the selection to attack the tree in a relatively small, short time frame so that they can together overcome the tree's defenses. And this attack synchrony is what contributes to outbreaks. And so aggregation, in fact, limits the efficiency of natural selection, and it allows for the persistence of extreme phenotypes, like some beetles that produce very little pheromone and some beetles that produce a lot of pheromone. So once that was done and Matt was convinced about my arguments, I found another postdoc with Therese Poland, who worked for the US Forest Service. And this was at the time when the emerald ash borer was a new pest. And a lot of the general natural history and ecology were unknown. So this is originally a pest from the Far East, and the questions where it is not a real serious pest, but in the US it was killing trees and causing a big problem. So we asked questions such as, how do these beetles locate these ash trees? Why are they so successful on North American ash trees? How do they locate mates? And do they use vision or olfaction for mate location? So we found, in fact, that these beetles preferred the North American ash trees, which had lower volatile emissions compared to European or Manchurian ash trees. And to find their mates, beetles could identify females before coming into physical contact. So we did a series of experiments where we blocked their vision and olfaction, and both. So you can see the beetles don't really have red eyes. We painted their eyes and found that, yeah, they can identify females before coming in contact with them through short-range olfactory cues. And once that was done, I got a permanent job at the Canadian Forest Service because, again, there was another pest species at work. This was the spruce budworm, which causes large-scale mortality of trees in eastern Canada. And it has deep endemic phases where you never see the insect, and severe epidemic phases where large areas of forest are completely consumed. So these extreme population fluctuations were of intrigue to ecologists and entomologists over several decades, with the questions as to what causes outbreaks, and actually now with the consequences of climate change, what are the implications that these outbreaks can have in the future. We found an interesting result when, over about a 10-year period, when we were working on the spruce budworm outbreak in northern Quebec. What actually triggers outbreaks and maintains them when at very low densities you hardly find the insect? So there was a theory that nitrogen is an important resource that stress trees produce that can actually trigger and maintain outbreaks in the long term. So we tested this theory by quantifying over a range of forest types how much nitrogen is in the soil during the course of the outbreak. We found that once the larvae start defoliating the trees, the needles and frass drop to the ground, the nitrogen concentration in the ground increases, and this is actually taken up by the trees. Even when they're severely defoliated, we found that the new foliage was, in fact, very, very high in nitrogen, and that this is actually stimulating the insects to do really well and to have more offspring, and contributes to sustaining the outbreaks. So it's like a self-sustaining mechanism once an outbreak is triggered in very mature stands. So what are the consequences of these outbreaks with climate change? So previous outbreaks occurred in Quebec much further south than the current outbreak. So you can see these are the spots from the early spots from the previous outbreak, and these are the early spots from the current outbreak. And in this area, the forest did not evolve to cope with these outbreaks. So we don't know what the consequences of these outbreaks will be this far north. And there's a secondary host species, which with climate change could become a more suitable host species for the spruce budworm. So black spruce is currently, there's a 2-week delay in bud burst, and this could change as the climate changes. So we predicted that there would be climate-induced narrowing of phenological mismatch with the secondary host species, that higher defoliation and mortality would occur in the boreal forest as a result of this. And once the black spruce forests are defoliated, we don't know what would happen once the canopy is opens up, and we don't know if black spruce will regenerate the same way that balsam fir can regenerate in the more southern forests. So we predicted that there would potentially be lower stand productivity, changes in regeneration patterns and nutrient cycling, and that overall there'll be altered ecosystem dynamics with nutrient poor habitats, such as the northernmost habitats, that would become less productive in the long term. So some of this work is still ongoing. The work is way far north, up to the 55th parallel, and I'm located down here in the south, in the middle of New Brunswick and Fredericton. And so we drive all the way, take a ferry across, and then drive several hours. I think the whole journey takes about 12 to 14 hours to work in these northern habitats. This is what it looks like. It's wild and beautiful, and it's very sparsely populated. We work with First Nations partners and with the Ministry of Forests in Quebec. And throughout the years, I've had several collaborators and technicians working with me to make my career rich and fruitful. I've worked with several graduate students as part of my research program, asking new questions about several pest species. I have collaborators in Europe, some of whom you might know. These were my close collaborators in Quebec for about a decade, and more students in Quebec. And my new research team at the lab in Fredericton, where I only moved a couple of years ago. So there are a lot of people who made this possible, and my career is quite interesting and exciting. And so I would like to encourage young women to pursue careers in forest science, because we can all do it, and it's fun. Thank you for your attention.
I lost my mic. Thank you very much, Deepa, for that very exciting presentation. Does anyone have a question? So while the people are thinking about it, I had one. You showed us that map of Quebec with those 2 outbreaks. I think one from '69 and the other one these recent years.
Yeah.
Is it the same population that outbreak and migrate there, or was it 2 different population? Or
it's a new population. So
okay.
That outbreak collapsed in the 90s.
Yeah.
And then a new outbreak started in 2006 in a different region.
Okay. And
yeah, so there's population cycles of 30 to 40 years. And so that outbreak began in southern Quebec. And actually, during the end of that past outbreak, insects were dispersing north, but the impact on the forests was pretty low because they were there towards the end of the outbreak, and the outbreak collapsed before there was a significant impact on the forest. But then this current outbreak, it started in those northern forests, and it's still ongoing there. So the impact on the forest is higher. Yeah, it's different populations. There's a period where it's endemic between the 2 outbreaks where you don't find the insect at all.
Okay.
I have a question. Great talk. I'm just curious if you could talk on the pheromone differences in the research that you did on the southern pine beetle. Did you find that pheromone production levels changed between sexes, like based off of who was the pioneer?
Yeah, so the different sexes produce different pheromones. So that's the
Okay.
female is the pioneer sex, and she produces aggregation pheromones, and then the males come in and produce more aggregation pheromones, and then they both switch to producing anti-aggregation pheromones.
Cool. And you said that
Yeah.
varies like based off of when they arrive at the tree as well?
Yeah,
like
so actually the early arrivers they would need to produce more pheromone, or they would just die.
Right.
And the middle beetles are the most successful in terms of producing offspring.
Cool. Wow. Interesting.
Yeah.
Thank you, Taylor, for your question. Do we have more questions? So just a bit of a technical question. When you looked at those beetle pheromone, where you only had a few that release a lot of pheromone and most of them didn't, did you quantify the quantity of pheromone from their gland or the quantity of pheromone they release?
Yeah, so there were different types of quantification we did. So for some of them, we aerated them in chambers.
Yeah.
So we would put them in logs, take them out, and aerate them in chambers. Some of them, we use another technique where we just dissected the hindgut and put them and extracted the hindgut in solvents.
Yeah.
So no matter what technique you used, there was still high variation.
Okay. Yeah, I was thinking if you just use aeration, you might not be sure if they are just not willing to release pheromone because, you know, the setup is wrong except for one or two, but
yeah, we also aerated them directly from like live standing trees. So we had this field set up where we took a pump and we attached the adsorbents on the trunk at the gallery, and we collected volatiles that way as well. So there were at least 3 or 4 different techniques we used.
Yeah.
And Brian Sullivan, who's my collaborator in Louisiana, he has all these contraptions, these mobile contraptions that you can take anywhere and set it up. Yeah.
All right. Do we have more questions? If we don't, please join me with another round of applause to all our speakers today. It was very inspiring. We were very lucky with all our wonderful scientists today. And Josephine, if you want to have the last word for this seminar.
Yeah, thank you so much for our speakers and for everybody that joined the session. Thank you for everybody, and I hope you enjoyed the last session that will be hosted in South Africa by our colleagues at FABI. And yeah, it was very fun. So thank you very much, everyone.